Hi, and welcome to Photosynthesis and Cellular Respiration project for Bio20 by Dakota Hawforth, or me. So first, we're just going to start off by describing what photosynthesis and cellular respiration really are, how they're connected, the processes, products, and reactants of each, and just how they are interconnected in our world. So firstly, all living organisms on Earth rely on the chemical processes of these two biochemical processes in one way or another. First, photosynthesis traps radiant energy from the sun and stores it in the form of glucose. Oxygen gas is created as a byproduct, which is used for other organisms, such as humans or any other animals, to breathe. Cellular respiration breaks down the chemical energy in glucose into ATP. Water and carbon dioxide are produced as a byproduct, which is why we breathe out carbon dioxide as human beings. And we also take in this oxygen for this cellular respiration to occur, which is what we'll get into later. So now we're going to be talking about photosynthesis in more detail, but just to break it down generally first, photosynthesis is one of the two biochemical reactions, cellular respiration being the other, that are essential for life on Earth to exist. There are three general steps to photosynthesis, such as light-dependent reactions, which encompasses two, and light-independent reactions, which encompasses the third one. The general equation for photosynthesis is 6 molecules of carbon dioxide, 6 molecules of H2O or water, and then energy or sunlight, and then which makes 1 molecule of glucose and 6 oxygen gas. So first, we're going to talk about our chloroplasts, which is where photosynthesis occurs, and are small organelles that are found only in plant cells, which is why only plant cells can produce photosynthesis. They are suspended in the cytoplasm, and they contain a green pigment chlorophyll, which aids and just is the site of photosynthesis. Now moving on to the first general processes of photosynthesis, which is the light-dependent reactions. So these light-dependent reactions encompasses stage 1 and stage 2 of photosynthesis. They occur in the chloroplast of each photosynthetic cell, and they include structures such as photosystem 2, which is found here, photosystem 1, which is located here, and the electron transport chain, which connects all of these in the membrane, and then finally our ATP synthase, which we'll talk about later. This last little complex here is just a protein pump where hydrogen is moved between the two. Now, first we're going to talk about stage 1, which is capturing solar energy and the electron transport chain of photosynthesis. And then, to begin photosynthesis, solar energy must be captured through the chlorophyll in photosystem 1, photosystem 2, to excite an electron which moves down the electron transport chain. Although as it moves, it begins to lose energy. This is where photolysis takes place and breaks down water molecules, which causes a buildup of hydrogen ions in the thylakoid membrane. And this little picture is about the photolysis of water. So first we're given H2O, which is water, which breaks down to be 2 hydrogen ions, 2 electrons, and half of 1 oxygen gas, because oxygen is a diatomic molecule and it cannot be found by itself. So these 2 electrons are put into photosystem 2, and then we are given ADP, which is reduced. So now moving on to our next stage of photosynthesis, stage 2, which is ATP synthesis. Now next, the buildup of hydrogen ions in the thylakoid is pushed into the stroma by the energy released by the ETC, or electron transport chain, in the first step. This is called chemiosmosis. It does this via the ATP synthase, and in the process it reduces ADP to ATP and NAD plus into NADPH. While photosystem 2 is performing, electrons are excited in a second photosystem called photosystem 1, which leads to similar events. The electron that leaves photosystem 2 is replaced by the electron that leaves photosystem 1. So in here, as you can see, there are a wide concentration of hydrogen ions in this top half. And then the ATP synthase, its job is to push these hydrogen ions through to satisfy this concentration gradient, which nature just does not like, which it always tries to satisfy. And then in the process of this, ADP is reduced to ATP, which creates energy. And then these steps described how this whole diagram is working. So we're starting here in photosystem 2, where the electrons are excited and passed down the electron transport chain to photosystem 1, which gives off energy and hydrogen ions in photolysis, passed along, and then excreted down here. And these hydrogen are going to be pumped through to satisfy the concentration gradient, which creates ATP. Now moving on to our a rather deeper explanation of the electron transport chain in chemiosmosis is now. The electron transport chain in the chloroplast is in the thylakoid membrane and is a chain in which electrons can be passed down to generate energy and is transported between photosystems, as what we've described before. And now chemiosmosis is not a structure, but rather the process of how the concentration gradient is satisfied, where hydrogen is high concentration on one side of the membrane and low on the other. Naturally, it's going to try to reach a state of equilibrium, and it does this by passing through ATP synthase while creating energy. Now finally, we can move on to our light-independent reactions, where these do not need light to occur. The Calvin cycle. The Calvin cycle uses both ATP molecules and high-energy electrons from light-independent reactions to finally form a molecule of glucose. Now, in a more general explanation, the Calvin cycle has 3 different steps. The first step, molecules of carbon dioxide react with RuBP to create 3-PGA, which is what we can see here. Carbon dioxide reacting with RuBP, catalyzed by Rubisco to create 2 molecules of G3P and G3P. And then in our next step, ATP activates these molecules of 3-PGA and NADPH to react with them to form molecules of G3P. As you can see, this PGA molecule and a NADPH molecule to create G3P. In our final step, the replacement of RuBP, 10 out of 12 of these G3P molecules, an additional 3 ATP must be recycled to replace RuBP molecules at the start for this reaction to be considered a cycle. So as you can see, 5 out of 6 in this are pulled along and back to replace these RuBP. But you see, this whole diagram only really shows what happens once. So this needs to happen twice to form a whole molecule of glucose, because 2 G3Ps form 1 glucose. Now to move on to our net equation, it uses 6 carbon dioxide, 6 RuBP, 15 ATP, and 12 NADPH to create 6 RuBP to replace the cycle, and 1 molecule of glucose. Now this may seem like a lot of ATP and NADPH wasted just to create 1 molecule of glucose, although we'll see later about how this actually is just a minuscule amount. Now, an overall diagram, which is similar to what we've had before, but shown in an actual cycle. 3 carbon dioxide is catalyzed by Rubisco with 3-RuBP to form an intermediate molecule which instantly breaks down into 3 PGAs. Now, this uses ATP, which is then reduced back to its uncharged form of ADP, and then is reduced by NADPH to make molecules of G3P. And then G3P are recycled to regenerate the cycle of RuBP back here, following it along, and this uses an additional 3 ATP, but then the 2 G3P remaining are used to actually form glucose. And see, as you can see here, this process happens twice. Now, we're going to move on to our overall diagram of photosynthesis. So then in this general chloroplast, there are both light-dependent reactions and the light-independent reactions of the Calvin cycle. So light hits the light-dependent reactions, where all this electron excitement begins, and then H2O is entered, it's broken apart by photolysis, and oxygen gas is released for the animals to breathe, as mentioned before. As you can see, the ATP produced and the NADPH produced are used in the Calvin cycle to create 1 molecule of glucose, and then recycles these ADP and NADPH back to the light-dependent reactions where they repeat this cycle. Carbon dioxide also enters the Calvin cycle so that glucose can be formed, because you need the carbon for the carbohydrates to be formed. And then glucose from there can either be starch or sucrose. Now, finally moving on to cellular respiration, our next topic. Now, just an overall breakdown of respiration like we did for photosynthesis is that cellular respiration is the second biochemical process performed by both plant/producers and animals or consumers. It is very important for converting glucose into usable energy. It is a catabolic pathway, meaning that it actually breaks down larger molecules into smaller molecules for energy, with many intermediate products and reactions. There are two different conditions it can occur under: aerobic and anaerobic, meaning that it occurs differently whether oxygen is present or not. This diagram is just an overall little flowchart of how each type occurs, which we will talk about next. Now, our first step of cellular respiration is actually glycolysis. Glycolysis is an anaerobic process, and it occurs in both types of cellular respiration, aerobic and anaerobic. Its purpose is to break down molecules of glucose into molecules of pyruvate for later oxidation, although it occurs in 3 steps. In our first step, 2 molecules of ATP break down glucose into G3P molecules. As you can see, glucose is a 6-carbon chain broken down using 2 ATP into glyceraldehyde 3-phosphate, or G3P. And then in our second step, G3P reduces NAD plus to NADPH, which generates 2 ATP molecules each with a net gain of 2. As you can see, NAD plus is reduced to NADH, and 2 ADP are reduced to ATP. So then pyruvate is formed, which is our final product described in the third step. The stage above shows how PGA, which would then become pyruvate, a high-energy molecule, is required for the Krebs cycle to occur. So 2-pyruvate are formed from this general process of glycolysis from this glucose molecule. Now, moving on to our aerobic stages of cellular respiration, there are 3. Although pyruvate oxidation must occur before the Krebs cycle can occur, because the Krebs cycle will not accept the pyruvate, which is created by glycolysis, because it must be converted into something called acetyl coenzyme A, which we will get into later. Now, this general diagram we will get back to afterwards. Firstly, pyruvate oxidation is like a precursor reaction that must occur before the Krebs cycle can happen, which is why it is only considered step 2A instead of step 2, and Krebs cycle being step 3. This process converts pyruvate into molecules of acetyl coenzyme A, which mentioned earlier, and happens in 3 steps. 1 carbon dioxide portion is removed from pyruvate. As you can see here, the carbon dioxide leaves. NAD plus is reduced to NADH. Coenzyme A is attached to the remaining 2-carbon protein. So as you can see, coenzyme A here, it forms acetyl coenzyme A with the acetyl group and the coenzyme group. And then this must happen twice because, as you can remember, 2 pyruvate were formed in the glycolysis from 1 breakdown of glucose. And then in step 2B, the main step is the Krebs cycle. It is an 8-step process in which the final product is also a reaction, which causes it to become a cycle. The purpose of this cycle is to transfer energy from organic molecules of acetyl-CoA into 3 molecules of ATP, 3 NADH, and an FADH2 molecule. CO2 is also created as a byproduct. Now, to explain this whole complicated diagram, we're going to start here up at acetyl coenzyme A. So first, it is transformed into all of its intermediate molecules, such as citrate, isocitrate, and etc., where NADH is created, a CO2 group leaves, another CO2 group leaves, and another NADH is produced. Now, ATP is also produced with all these intermediate molecules and all these intermediate processes, because I'm not going to explain all 8 steps of this cycle. Then the FADH2 is also created, and finally another NADH, where the alloxylacetate is its final product that will be continued back in the cycle. So that's the Krebs cycle, and moving on to the ETC and chemiosmosis. The electron transport chain and chemiosmosis of cellular respiration are actually very similar to the ETC and chemiosmosis from photosynthesis, although they occur in aerobic cellular respiration, and it is the most valuable and final step where most ATP is produced. The electron transport chain is actually in the inner membrane of the mitochondria this time, and not the thylakoid membrane of the chloroplast, because, as mentioned before, the organisms that perform cellular respiration actually do not have chloroplasts, as they only occur in plant cells. And the movement of hydrogen ions being pulled through the ATP synthase complexes reduce ADP to ATP, although in prime conditions, up to 34 ATP molecules are produced. This is not always the case, as oxygen is not always ready. So, moving on again, we're just going to explain this general cycle. So as you can see, glycolysis begins this process over here, where it converts glucose into 2 pyruvate molecules, which then also creates an ATP, or 2 ATPs rather. And then they move to the citric acid cycle, which is also known as the Krebs cycle, is what we know it as. And then all the processes happen that produces ATP, and then it transfers NADH and FADH2 to oxidative phosphorylation, which we know as the electron transport chain and chemiosmosis, where up to 34 ATP are produced here, as you can see, which provides most of the energy for the cell. Moving on to the anaerobic stages of respiration. So there are only 2 steps of anaerobic cellular respiration, the first being glycolysis, similar to aerobic cellular respiration, because glycolysis is actually an anaerobic process and does not require oxygen to occur. So then our second step that are described is fermentation. And there are actually 2 types of fermentation, and they're named according to the byproduct that they produce. So alcohol fermentation produces ethanol, also known as alcohol, and then lactic acid will produce lactate or lactic acid. So this chart we will come back to after we look at different types. So alcohol fermentation is our first step. Now, just to give an overall description of fermentation, it is a metabolic process that produces chemical changes in organic substrates, or in this case, the extraction of energy from carbohydrates in the absence of oxygen. Alcohol fermentation is performed by many bacteria, and the byproduct of it produces alcohol. This allows NADH to be recycled so that glycolysis can be continued. Carbon dioxide and ethanol are released as waste, and the 2 ATP produced by glycolysis are sufficient for the organism or bacteria to meet its needs. And this is just a short little flowchart that shows the pyruvate and how it is converted into ethanol or alcohol, with the carbon dioxide leaving it as waste. And then here the NADH is converted back to NAD+, its uncharged form, which then it returns to the glycolysis for it to be recycled. So that would be alcohol fermentation. Moving on to lactic acid fermentation. In this type, lactic acid is produced as a byproduct. As we mentioned, our muscle performs this when it has limited access to oxygen, and is what causes muscle cramps. NADH transfers its hydrogen atoms to newly renewed pyruvate for the recycling of NAD+, allowing glucose to continue. So this chart here actually shows glycolysis and fermentation, so the entire process of anaerobic respiration. So here, glucose is transferred into 2 pyruvate through glycolysis, and the 2 pyruvate is fermented into 2 lactate or lactic acid. So here this shows how the 2 NAD+ are converted to 2 NADH in glycolysis, and then also these are converted back in fermentation, so it's using it up, where then it can be recycled back through this arrow to NAD+. So moving on to our overall diagram of cellular respiration. So now here we're going to start at the very beginning with glucose, which is transferred into pyruvic acid because of glycolysis, which is an anaerobic process that happens both in aerobic and anaerobic respiration. The net gain of ATP from this process is 2. And then if oxygen is present, it is moved on to the Krebs cycle, and then oxidative phosphorylation, also known as the electron transport chain. This produces a total of 36 ATP plus the 2 net ATP from before. If oxygen is not present, then fermentation occurs. And there are 2 types, lactic acid, which happens in animals, and ethanol or alcohol, which is in yeast and bacteria. Now that is cellular respiration. Now finally, we're going to explain the connections between both of these essential processes. So we're going to explain the biochemical interdependence of these 2 processes. All organisms require a constant input of energy, although they achieve this in many different ways. Most organisms perform photosynthesis, cellular respiration, or both, with few exceptions. Photosynthesis's main goal is to store energy, while the main goal of cellular respiration is to release this energy that's photosynthesis stored. These two processes differ in which organisms perform each, the reactants and products of each, where each occurs, and the major steps of each. Now we're going to move on to the similarities and differences of each. Now, photosynthesis and cellular respiration are complementary processes that sustain life on Earth. Together they regulate atmospheric concentration of oxygen gas and carbon dioxide. The two's products and reactants work inversely so that photosynthesis uses the products of cellular respiration and vice versa. Cellular respiration also occurs differently whether oxygen is present or not, while photosynthesis is used to make this oxygen. It also does not require oxygen, so it can happen no matter if oxygen is present or not. Photosynthesis requires light to occur, while cellular respiration does not have the chlorophyll that requires the light to excite the electrons. Both have an electron transport chain that performs chemiosmosis, which is what we're going to elaborate on next. So the electron transport chain and chemiosmosis have many similarities in the two processes, but also few differences. Photosynthesis, ETC, is in the thylakoid membrane of the chloroplast, and the ETC reduces NADP+ to NADPH. On the other hand, cellular respiration's electron transport chain exists in the inner membrane of the mitochondria, as we mentioned earlier, and it reduces NAD+ to NADH and FAD+ to FADH2. Finally, the electrons in photosynthesis are excited from the sunlight, as you can see here, being absorbed by the chlorophyll in photosystem 2 and photosystem 1. Although the electrons in cell respiration are donated from NADH down here and FADH2. And after we have explained these key differences, I think it is time to move on to the overall diagram of both cellular respiration and photosynthesis. So here, this can be a lot to take in. We are going to start here with oxygen. So here, the oxygen, when it is in this precipitated form of H2O, the light-dependent reactions will take it in and it will perform photolysis on it, breaking it down into its hydrogen and its oxygen. See, the oxygen is released here again for just as a waste product, and then here, as it's a waste product, it's going to be put into the electron transport chain and then used to form H2O again with the excess hydrogen ions. So here it's going back and it repeats the cycle. If we're going to take a look at the carbon dioxide between the two, we're going to look at how it enters the Calvin cycle and it's broken down into carbon and its oxygen. So the carbon is what's going to help us form these carbohydrates, also known as glucose, which then transfers along here and is used in glycolysis to break down into pyruvate and start the Krebs cycle. If we backtrack, this carbon that was moved all the way, and because pyruvate is a 2-carbon chain, it's going to enter here and then it's going to excrete these carbons binded to oxygen as carbon dioxide, which then will enter the Calvin cycle again, like mentioned before. So all of this atmospheric regulation is in a huge chain. All of life's processes work in a cycle. This is going to repeat over and over again until life just ends on Earth. And that is just how photosynthesis and cellular respiration are intertwined and interconnected. So moving on to the end of our slideshow, I'd like to thank you for listening, and I hope that you learned a lot about cellular respiration and photosynthesis. So, bye. In this case, the extraction of energy from carbohydrate. Okay.